Right, hello everybody. We are back here finishing up chapter 12. So we just finished covering the cell cycle, and now what I want to do is cover cell cycle regulation. And cell cycle regulation is going to be controlled really by those three checkpoints that we talked about briefly in the last video. So if you remember, our cell cycle looks something like this. And remember, we had G1 or GAP1, we had the S phase, we had G2, and then we have the M phase. So there are three checkpoints throughout this cell cycle. And when I say checkpoint, I mean that's a point that the cell is actually going to stop. It's going to sort of check a few things different for each checkpoint and decide if that cell is going to be able to move forward in a healthy manner. And when I say that, I'm mostly talking about stopping uncontrolled division. So the reason that we do these things, the reason that we have checkpoints, which we have one here at the end of G1, we have one here at the very end of G2, and then there's another one midway through mitosis during metaphase. And the reason we do these checkpoints is we want to make sure that our cells that we're producing, when we do split one cell through mitosis, we want to make sure that they are identical and that they are healthy. So if these checkpoints fail, you can get things like cancer, which is just uncontrolled cell proliferation. So these checkpoints are called the G1 checkpoint, the G2 checkpoint, and the M phase checkpoint. So just to really briefly, and we'll get into all this stuff on the whiteboard in a second, but just to go over sort of what each checkpoint checks for, I'll do this down here. The G1 checkpoint is really, really importantly going to check the DNA for any damage. That's really the key one. That's the one to remember for that G1 checkpoint. It will also make sure that the cell is growing properly, so growth check as well, but mostly we're going to be looking that that DNA is healthy. Our G2 checkpoint is the one that's going into mitosis. Remember, so it's at the end of G2. You might also want to call this maybe the G2M checkpoint because it's going into mitosis. And that is just going to really, really importantly make sure that the cell is ready to divide. So it wants to make sure before we get in that process of mitosis that the cell is going to be able to do that properly. And then the last one is the M checkpoint, which takes place during metaphase. And if you remember, metaphase is where all those chromosomes are going to line up in the middle along the metaphasic plate like that. And that's what this is going to check for. The M phase checkpoint is going to make sure that daughter cells are receiving the proper amount of chromosomes. So daughter cells are receiving proper chromosomes. I guess I don't need to write it out, I can just say it. Another way to think about that would be making sure that the sister chromatids in anaphase are going to split properly. Because what we don't want to happen is we have a cell like this, and maybe these spindle apparatuses improperly attach, and maybe something like this happens, and it's going to pull it in all kinds of weird ways, and you might end up with two daughter cells that have different amounts of DNA, and that's how you get things like incorrect ploidy numbers, so incorrect N numbers. This would be like how you would get trisomy, for example. The most common example would be trisomy 21, which is trisomy of your 21st chromosome, and that's what we call clinically Down syndrome. Alright, so now that we've just kind of briefly covered what those checkpoints are for, it's time to get into sort of the meat and potatoes of everything, which is going to be how this works. So we're going to go through this one at a time. So really, and I've already written them up here, there are two types of genes that are sort of battling it out constantly when it comes to cell regulation. You have your proto-oncogenes, which promote cell growth and division, and that's kind of like hitting the gas pedal on your car. And then you have tumor suppressor genes, which are all going to be in red, and tumor suppressor genes are like hitting the brakes in your car. They're going to stop the cell from proliferating. They might even kill the cell if it determines that the cell is unhealthy and that DNA is going to be a problem. Sometimes it'll just kill the cell, and that's called apoptosis, which we'll get into a little bit later. So I'm going to start with sort of the two stars of the show. I got erased a little bit. But these first two genes here are really important. So this is your cyclin gene and your CDK gene. And they might have a bunch of different names, but all you need to know is that they are going to produce cyclins. And cyclins are just these little transcription factor-esque proteins that are going to be able to turn on that are going to have some kind of like effect on other things, which we'll get into later. And it's also really important to know that these cyclin genes are stimulated by growth factors. So growth factors are going to come into the cell or be produced by the cell, and they're going to stimulate that cyclin gene to start making cyclins. Now, quick side note, you see here we have 4 kinds of cyclins. We have cyclin D, cyclin E, cyclin A, and cyclin B. And those 4 types of cyclins are going to be more active at different stages in the cell cycle. And I have that on this graph over here, which I'll go over after I go over CDK real quick. So CDK means, what CDK stands for is cyclin-dependent kinase. All right, and you remember anytime you see the word kinase, so let's write that out actually. So this is a cyclin-dependent kinase. And you may remember anytime that we see the word kinase, it means it's going to be phosphorylating something. So we know at some point that this CDK is going to add a phosphate group to something, but we will get to that later. So now that we know cyclin and CDK, it's important to understand how they work together. So these cyclins are produced in response to certain growth factors at different stages in the cell. But this CDK is sort of always being produced. And these CDKs and these cyclins really want to combine. They have a very high affinity for each other, and they want to bind and form. Ooh, I drew this poorly, but this is the cyclin and this is the CDK, and this is going to form the cyclin CDK complex. Now, another word that you might see to determine a certain type of cyclin CDK complex, you might hear it called a maturation promoting factor, which we shorten to MPF. And MPF is very specifically going to be cyclin B, but we will get into that again in this graph here. So we have these cyclin CDK complexes now. And I'm going to save this graph for the very end. We're going to go through all this first, and then we'll get to that. So now that these cyclins and CDKs have combined into a cyclin CDK complex, what is it that they're doing? Well, they're actually going to inhibit the work of another gene. So the gene that they are going to be inhibiting, the protein that they're going to be inhibiting is this RB guy. And this RB stands for retinoblastoma protein. And what's up here as well, it's the retinoblastoma gene. And you see it's in red, which means that that is a tumor suppressor gene, which means that this retinoblastoma wants us to stop the cell cycle. It wants to put an end to proliferation. So how does it do that? We'll get back to the cyclin CDK complexes once we go through this. So there are these transcription factors that just are sort of produced and floating around in the cell, and they're going to be called E2F. You don't need to know what it stands for. It's just E2F. And E2F, very simply put, stimulates cell proliferation genes. So the job of E2F is to start proliferating the cell. So E2F wants the cell to progress through the cell cycle. It's hitting the gas pedal, and it does that by causing transcription or activation of these cell growth and cell proliferation genes, which you don't need to know what specific ones. Just know that E2F stimulates proliferation genes. And it's really good at doing that, but what this retinoblastoma protein is going to do is it's going to bind right here to E2F, and when retinoblastoma binds to E2F, it's going to stop it from being able to activate these proliferation genes. So once retinoblastoma binds here, we can no longer activate these cell proliferation genes, which means we can no longer move forward with the cell cycle. So, how do we get rid of this retinoblastoma? That is the job of the cyclin CDK complexes. The cyclin CDK complexes are going to come over here and they're going to phosphorylate. Remember, because we have a kinase, so it's going to phosphorylate this retinoblastoma protein. And when it phosphorylates that retinoblastoma protein, it forces it to detach from E2F. And then once it is no longer attached to E2F, there's no longer anything stopping E2F from going ahead and starting cell proliferation. And so now, this retinoblastoma has a little phosphate attached to it. And with that phosphate attached to it, it cannot bind. It cannot bind to E2F anymore. So that's what cyclin CDK complexes do. It essentially stops the thing that's stopping cell proliferation. So just quick vocab check, just to make sure we're all on the same page. We have cyclins, which are created when the cell gets stimulated with growth factors. And we have cyclin D, E, A, and B. And then we have our CDKs, or cyclin-dependent kinases, which are going to combine with the cyclin to create a cyclin CDK complex. And those are going to phosphorylate retinoblastoma. And what retinoblastoma proteins want to do is inhibit E2F. And E2F wants to start proliferating the cell. I think it's helpful to think of this process backwards sometimes a little bit, because if we want the cell to progress, we know that we want cell proliferation genes to be active. So then I think, okay, what activates cell proliferation genes? E2F. And I go, okay, what can affect E2F? Retinoblastoma can affect that E2F and inhibit it. And then I go, okay, well, what interacts with retinoblastoma? Cyclin CDK. And this is all going to come full circle because we are eventually going to get something that's going to inhibit cyclin from binding to CDK as a result of activation of this guy down here, which is your P53 tumor suppressor gene. Everyone just round of applause for your P53 tumor suppressor gene, because it is single-handedly stopping most of the cancer in your cells. It is an extremely efficient gene at sensing DNA damage. So, let's say that we have a cell and there is some really bad DNA right here. So this is bad DNA that if we allow this cell to divide, this DNA is going to cause that cell to become cancerous and divide uncontrollably. So you have these other like genes and enzymes, ATM and ATR. You don't need to know that, but just know that they sense DNA damage. They proofread this DNA going into the S phase. So if we're going into the S phase, we know we're probably dealing with that G1 checkpoint. So that's where P53 is going to be very active. So this P53 is then going to be stimulated by damaged DNA. And this P53 is going to go, okay, hold on a second. I don't want this cell to divide. It would be unsafe for us to divide. So P53 is then going to go ahead and activate a couple sets of other genes, and we're going to talk about 3 categories. So the first thing that p53 can stimulate is a DNA repair gene. And what a DNA repair gene is going to do is it's going to produce these little enzymes. You don't need to know who they are, but they can actually go back to this DNA and they can try to fix it. They can try to, maybe we have something that's mispaired. Maybe we have an A that's trying to pair with a G instead of a T, right? It can go back, look at that, and go, hey, wait a second, that's not right, and it can fix the DNA, and it can sort of heal this stretch of DNA, and then p53 will check it again, and then maybe p53 goes, okay, we're good then, we can continue with the cell cycle now that this DNA is repaired. But maybe this DNA repair enzyme doesn't do the job, and we are still getting signals to p53 to go ahead and stop the cell cycle, because we still have damaged DNA even after the work of our DNA repair enzymes. Well, then p53 is going to start stimulating your Bax genes. Bax genes, you don't need to know the name Bax gene, but just know that there are these pro-apoptotic genes. And what apoptotic genes do is they make apoptosis happen. You can think of apoptosis as the cell popping itself. It's just blown itself up. It's going to essentially crunch all this DNA and basically scrap it, and then it's going to let the cell just rupture and let all of the good stuff, like maybe we have some good mitochondria in the cell, we want those to go out. And be used maybe by other cells, but we don't want this cell's DNA to divide. So apoptosis is just programmed. I wrote that really bad, but that says programmed cell death. And apoptosis, all I want you to know really about it, other than what I've said, is that it is controlled by enzymes called caspases. Caspases are basically just responsible for breaking stuff down that we don't want and then lysing the cell membrane, or just cutting the cell membrane open and letting the contents spill out. So the last thing that the p53 gene might do, and this is going to bring it all full circle, is it can interact and activate with these other P genes. So maybe it's P15, P14, there's a P19, there's a P27, there's a P51, there's a P65 maybe. There's a ton of these. I don't care. Just know that one thing that P53 can eventually do is inhibit cyclin from binding to CDK. So this is as a result of P53, it can inhibit cyclin CDK binding. And if we sort of track that and look what happens, if this is inhibiting the cyclin CDK binding, then this is never going to happen. There's never going to be a joining of the cyclin and CDK, which means we're never going to be able to phosphorylate this retinoblastoma protein. And if there's no phosphate on that retinoblastoma protein, then that retinoblastoma protein can go ahead and inhibit E2F. And if it inhibits E2F, it inhibits proliferation. So that is sort of the molecular processes involved with cell cycle regulation. I'm going to clean us up a little bit. Take a picture now if you'd like. It's all very beautiful now. All right. So now we are going to talk about this graph and talk about our different kinds of cycling. We're not going to go too crazy in depth, probably just a few minutes, because that's more cellular biology, so that's by 336 if you ever take that. But, so remember we had 4 cyclins. We had D, E, A, and B. I always think of it as Deeb. I don't know why, but it spells Deeb. And the reason I write it this way is because this is sort of the order that these cyclins arise. And I have them written down on this graph. I'm going to kind of space them out so you can see which each one is doing. So cyclin D is the first one, and cyclin D is sort of present throughout the whole cell cycle. Cyclin D looks kind of like this. So that's just, it's present throughout the whole cell cycle, and then it, ends at the end of the cell cycle, and then will arise again at the beginning. So cyclin D is not super relevant for this class. It's very important. It does a lot of great stuff. Just know that that's the one that's sort of always around. But the next one is cyclin E. We need more space here, so we'll do A over here and B here. But cyclin E is going to arise at the beginning of the cell cycle, and then it's going to spike way the hell up really fast and really early, and then it's going to sort of diminish out towards the end. And conveniently, right here is your G1S checkpoint. So cyclin E correlates to the G1S checkpoint. Cyclin A will sort of begin arising a little bit into the G1, so it's going to be like, if this is the G1S checkpoint, cyclin A will start about here, and it'll spike way the hell up, and then sort of degrade, very conveniently again, at the G2M checkpoint. So cyclin A correlates to the G2M checkpoint. And cyclin B is one that I'm going to talk a little extra about because it's a special guy. Cyclin B is going to start synthesizing right at S phase. So right when S phase starts, we're going to start making cyclin B, and it's going to very, very slowly accumulate. But then once we pass once we begin mitosis, so this is the M phase start, it is going to spike up incredibly fast, and then it's going to degrade pretty fast too. And right here is going to be metaphase, which is where our M checkpoint is. So cyclin B correlates to the M checkpoint. And next up, I just want to talk about CDK levels. So CDK levels are at their peak and then very, very quickly degrade at each checkpoint. So we have our G1S checkpoint, our G2M checkpoint, and our M checkpoint. Cyclin is going to accumulate, fall off very fast, accumulate, fall off very fast, accumulate, fall off very fast, right at these checkpoints. And that makes sense when you think about it, because if we have more cyclin, we need more CDK, right? Last thing that I want to talk about, and this will be really quick as well, is just cyclin B and why it's special. So cyclin B plus CDK equals a maturation promoting factor. And the maturation promoting factors are controlling the M checkpoint. And all I really want you to know about these MPFs is that they are going to be controlling that M checkpoint, which means they are making sure that chromosomes line up properly, and split properly. And then just the other thing that I think is important to know is that these MPFs get degraded in anaphase, and their degradation, the degradation of these MPFs is actually what's sort of going to signal anaphase to happen and split, because they're going to get split by the anaphase promoting complex. So that's basically it for cell cycle regulation. Trying to think real quick if there's anything else. Not that I remember. but that's a very good outline. So I will see you guys for chapter 13, which is meiosis.